Office hours Thursday, 2:10 to 3 o'clock. I have to be somewhere that ends at 2 o'clock. It takes me about 5-10 minutes to get to the room. So 10 after 2 to 3. Also, you can always ask me questions by way of email. And there'll be more of that as we get closer to the end. But certainly feel free. Today we're going to do certainly male reproduction, spermatogenesis, and hormones negative feedback. And we'll get a good chunk into female reproduction. I'm sure we'll get to oogenesis, and we'll probably get to ovum development, maybe not the menstrual cycle today, which we'll pick up Friday. So first a review from Monday. Know the difference. Insulin stimulates the use of glucose from the blood, so it brings down glucose. Those of you who have eaten breakfast this morning are undergoing insulin receptor action as I speak. Those of you who did not eat breakfast this morning are undergoing glucagon receptor action to produce glucose in your blood. Those two hormones work by fundamentally different yet very important mechanisms. If you know these two, you know a lot about protein hormones. So insulin receptor action, as reviewed, the hormone binds the receptor clusters. In that family of receptors, they can also dimerize. You don't need to distinguish clustering from dimerization. Same basic concept. The receptor interactions activate the tyrosine kinase, which is in the receptor itself, within the structure. So these receptors are enzymes, and a very important class of enzymes called tyrosine kinases. In the case of insulin receptor, the other subunit of the receptor is phosphorylated out of tyrosine. In addition, there's a really important substrate called IRS, insulin receptor substrate. So the name gives you a hint what it is. And that is a heavily tyrosine-laden protein. It gets tyrosine phosphorylated between 5 and 15 places. And as a result, cell signaling molecules bind to the receptor or bind to IRS1, which we talked about. IRS1 basically represents an extended platform of the receptor. So it allows many more signaling molecules of different types to bind. And this interaction, this protein-protein interaction, activates the cell signaling molecules. A lot of these themselves are enzymes. You don't really need to know what they are. A lot of them kick on other pathways that are very, very important in the cell. And together, I'll call it, in the case of those of you who ate breakfast, causes storage of glucose in the glycogen and the use of glucose, glycolytic pathways and so forth. Okay? That's a fundamentally important protein receptor mechanism of action. A lot of growth factor receptors work similar to this. A lot of, in fact, they're involved in growth, a lot of growth factor receptors, and certain kinds of anti-cancer drugs, new wave anti-cancer drugs, are focused on different parts of this generally similar pathway in growth factor receptors. Diabetes. 10% of diabetic patients are type 1. That's the destruction of the beta islet cells. That's through autoimmune diseases, autoimmune response, and you lose insulin production. You can just simply give those patients more insulin, and you can balance what's going on. Type 2 diabetics, these are more adult onset diabetics. You have problems with the receptor, and you have problems with the cell signaling molecules. There's many cell signaling molecules. A lot of these are affected by fat content, exercise, good eating habits, et cetera, et cetera, et cetera. So that's why adults have this adult onset diabetes. It's due to changes in the expression and functionality of these cell signaling molecules and receptors themselves. There are specific mutations. There is a type 2 diabetic patient. There's a class of them that have a defect, or mutation rather, which causes a defect in the tyrosine kinase activity. So the tyrosine kinase is almost dead. So the hormone binds, the receptors cluster, and you do not kick on the phosphorylation events, and those patients are diabetic, telling you therefore the importance of the phosphorylation and the binding of the signaling molecules. Okay, there's not too many of those patients, but there's a couple very clear-cut cases that have been characterized. Any questions on this? It's a fundamentally important receptor mechanism of action. You all good there? Okay. And those of you who ate breakfast, all this is going on at the moment. For those of you who did not eat breakfast, you're into glucagon receptor action. And that's how you produce glucose. This, again, is by a fundamentally important receptor mechanism. And that is these receptors have 7 pass transmembrane parts of the, go through the membrane 7 times, and they're bound to a G protein that in its inactive state is bound to GDP. It's a sequence of events. Hormone binds, you have a GTP-GDP exchange that we talked about. You uncouple the GTP-G protein from the receptor. There's just enough of a subtle conformational change that the G protein no longer has much affinity for the receptor, but it does have affinity for adenylyl cyclase, which converts ATP into cyclic AMP. Cyclic AMP goes into the cell, binds to the regulatory subunit of the inactive protein kinase A, which is a serine-threonine protein kinase. You uncouple the two. The classic example of the regulatory subunit being the inhibitor of the catalytic subunit. A lot of examples of this in biology where one subunit is an inhibitor of the other subunit. There's also examples the other way. The catalytic subunit then goes into the runs amok in the cell, and the catalytic, when it goes into the cytoplasm, triggers a reaction due to phosphorylation, converts glycogen back down to glucose. You secrete out the glucose. In addition, it goes into the nucleus, phosphorylates a specific transcription factor, which then triggers the expression of gluconeogenic, the name gluconeogenic, production of glucose genes. So genes that produce more glucose. Together, you produce more glucose in your blood. And those of you who have not eaten breakfast, that's going on right now. There's an off switch, and that is the G protein within the structure of the G protein is a GTPase. It's like a fuse or an off switch. And the high energy phosphorylated group gets kicked off. You go back to the GDP state, which is the inactive state. You now have affinity for the receptor, and you shut down the system. Does everybody see that? Question?
Yeah, I have a real quick question. Earlier in the week, you talked about how...
You mean Monday?
Yeah, I think it might have been...
Early in the week? Okay. I'm joking. Late last week. Okay.
You talked about how glucose, when it's low, you get a hormone in the alpha.
Yeah.
Where does that fit in all this?
Oh, where does the glucose homeostasis fit in? When glucose is low. Question is, how does glucose homeostasis fit in? When glucose is low glucose levels triggers the alpha islet cells to produce glucagon, which is the hormone that binds the glucagon receptor. When glucose is high, triggers, in this case it's a little better known, triggers a depolarization of the beta islet cells and calcium influx, and you get production of insulin. So when those of you who ate breakfast, your insulin levels, glucose levels are up, therefore you produce insulin, you trigger the pathway over here. Those of you who did not eat breakfast, you produce glucagon, you trigger these pathways. Okay? So this is the mechanism of action of how these hormones work. You'll see in human reproduction, a lot of the protein hormones we talk about in human reproduction work by the 7-pass transmembrane receptor. Although because of the nature of the cells, you kick on reproductive events and not glucose events. Okay, the targets are different inside the cell, but there's some general similarities to the mechanism. Okay? So again, the off switch is this GTPase on the G protein, and cholera produces a toxin called cholera toxin, which covalently modifies the G protein in a way that keeps it on at all times. And so therefore you produce a lot of adenylyl, you make active adenylyl cyclase, you make a lot of cyclic AMP, and this is connected to water balance and loss of water. And therefore one of the problems with cholera is dehydration. And this is part of the mechanism of what's going on with that. Okay? Questions on that? And then finally, yes? Well, what would cholera toxin be doing? Not really a hormone antagonist in the way we're defining it, because it doesn't bind the receptor. It does muck up the receptor signaling pathway. And there's a lot of examples in medicines as well as disease states that in fact affect the cell signaling components. You could argue that it prevents glucagon from working. And you could make an argument that's an antagonist. But we're going to use the word antagonist only in the sense that it binds the receptor directly and prevents an agonist from working. Okay? There's a little bit of semantical argument you can make a lot in this case. In this case, just know that it keeps active a cell signaling molecule. Very good question. Yes?
By what mechanism is GTPase turned on?
By what mechanism is the GTPase turned on? By the, very good question, by the presence of the GTP, which is its substrate. The G protein is a GTPase. In the GDP-bound form, right, there's no substrate around. So it's like a fuse. The longer you keep this fuse on, the more cyclic AMP you have. Also, you can turn down cyclic AMP by phosphodiesterase. You can get rid of it. You drink coffee, you keep it around longer. Okay? Good questions. Other questions. Know the difference between these two mechanisms. I consider these fundamentally important in the sense, you understand this, you understand a big chunk of protein hormone receptor biology and how all these hormones work. All right?
What's the light switch again?
What's the what?
Light switch?
No light switch. The fuse. I call it a fuse. If this GTPase is really active, what would you predict? I mean, really active. You predict that you completely get rid of the GTP-bound form, right? Because you really convert it very fast. And there are certain oncogenes that have G proteins, not related to this G protein, but do other things, that have very fast, GTPases that keep this off. Okay, so again, you have on and off switches. In biology, turning something on is as important as turning it off. It's a balance. And as soon as you muck around with that balance, you get either good medicines or disease states, depending on what side of this thing you're thinking about. And a lot of meds that you guys will think about in med school, in fact, hit these kinds of pathways. Okay? Questions? All right. So, human reproduction. And I'm going to stick strictly with human reproduction on this. And in humans, it's considered sexual reproduction in the sense you've got two haploid gametes come together, and you get a diploid zygote. We'll see what some of this means in a little bit. For the males, which we'll talk about first, the gametes are spermatozoa. Females, it's actually a bit more complicated in that females do not make a gamete cell. What they make is a gamete nucleus. And you'll see what the complication is. That is haploid. They do not make a gamete cell, but a gamete nucleus. And as you'll see also, females make a potential gamete about every 28 days. And we'll see how that fits in. Now, the reproductive organs, the males, the testes, and the females, the ovary. And the reproductive organs have two very important functions. One is to produce the gametes. So in the case of the males, you make gametes. Very important to stress here. In the case of the females, you make potential gamete nuclei. You don't always make real gametes or haploid nuclei. You'll see why that is when we get to that point. The other thing that these guys do, I'll bring this board down in a second, is they make reproductive steroids. They make the key steroid hormones that we will be talking about. I'll leave this up here. For the males, we use T for testosterone, DHT for dihydrotestosterone. There's a critical difference between these. And E for estrogen, and P for progesterone. Well, basically, of the steroids we will talk about testosterone and dihydrotestosterone for males, and for females, we'll talk about testosterone, estrogen, and progesterone. Okay? So those are the main functions of the reproductive organs. Okay? So let's now start talking about male spermatogenesis. Spermatogenesis is constant. And the reason it's constant, as we'll get to, is because it involves a hormonal control mechanism of negative feedback. Very important negative feedback system. Remember we talked about negative feedback keeps a constant level of a process or a molecule. Okay? In addition, there's three general classes of cells. You have spermatogenic cells, and you have two types of accessory cells. We'll see how this all fits in. You got Leydig cells, which convert cholesterol, COL is cholesterol, to testosterone. And you have Sertoli cells that produce testosterone into dihydrotestosterone. Okay? Please note what these accessory cells do. We'll talk about how they're located near each other in a second here. Because for females, you're going to see similarities and differences of this particular comment. All right? So, where does spermatogenesis all take place? It all takes place in the seminiferous tubules in the testes. Tubules within the testes. The way to think about this is it's a series of tubes that are all interconnected. Think of it as a big plate of spaghetti. And that's kind of what these tubes look like, all interrelated, all intertwined, and so forth. And if we look at a cross-section of one of these tubules, we get the following. Okay, this is a cross-section. This is the basement membrane. Outside the basement membrane is the Leydig cells, which again converts cholesterol into testosterone. Inside the basement membrane are the Sertoli cells. Here's one, here's another one. And in between the Sertoli cells are the spermatogenic cells. Okay? So the Leydig cell is in contact with the basement membrane. The Sertoli cells are in direct contact with the spermatogenic cells. Okay? And I do it in a way where these Sertoli cells are kind of touching each other. What happens on the outside of this cross-section, the outer rim of the cross-section, is different than what happens on the inner part of the cross-section in terms of what goes on with these cells. Okay, questions at this point, because we're going to just complicate this ever so slightly here in a second. Just again, think of the anatomy. There's some of these spermatogenic cells on this outer periphery of the cross-section, others are on the inside. Yes? Is that like what? Yes, this is a seminiferous tubules. It's like a big plate of spaghetti. And you take one of those pieces of spaghetti and cut it in cross-section, so it's a cross-section. As you can imagine, there's lots of surface area of seminiferous tubules. And therefore, what I talk about for one cross-section is going on, multiply that by a lot. Okay? Very important to see. So, this upper board is going to be what goes on in this upper part, this outer part of the cross-section. Then the lower board, I'm going to put in what goes on the inside. Because there's fundamentally two different things going on. So let me just put in the Sertoli cells first. Okay, here's one Sertoli cell. And here's the other one here. Let's see. And where the cells touch each other is called the Sertoli bridge. There's one Sertoli cell, here's the other. So what I've blown up in higher magnification is basically this part right here. Okay? And this is the basement membrane. Out here. What is being constantly produced is a type of cell called spermatogonia. Okay, spermatogonia. And through mitosis, you make more spermatogonia. Spermatogonia are diploid. And what does diploid mean? 46 chromosomes, right? And 2 cDNA content. Does everybody have that from earlier, more or less? So you have 46 chromosomes and then 2 cDNA. These are true diploids. Diploids refer to chromosome number, but I call these true diploids because they have the correct amount of DNA, 2 cDNA. Okay? What happens then above this bridge is a DNA duplication. And you make a primary spermatocyte. Let me put this. I'll put it up here in a second. This primary spermatocyte is 46 chromosomes but 4 cDNA. So you duplicated the DNA, but you have not doubled the amount of chromosomes. These are basically bigger, thicker chromosomes. So what hits the top of the Sertoli bridge is a cell that is 46 chromosomes but 4 cDNA, because it undergoes a DNA duplication. Questions at that point? You guys had mitosis meiosis earlier, right? Okay, now we're using it for something. Okay? So this primary spermatocyte then goes through the bridge. So that primary spermatocyte going through is this guy right here. Okay, that's what goes through this connections between the Sertoli cells, is the primary spermatocyte. This then undergoes the first meiotic division of the DNA. And what you produce is 2 secondary spermatocytes. Because it's a meiotic division, this guy has 23 chromosomes but 2 cDNA. It is haploid in terms of chromosome number, but I personally do not call that a true haploid because it has too much DNA. Then these secondary spermatocytes undergo the second meiotic division of DNA, and you get 4 true haploids called spermatids. I'll put this board down in a second. And the spermatids are 23 chromosomes cDNA. I'll put this down a second here. And then each of these spermatids undergoes a differentiation process where they make these immature spermatozoa, which are also 23C. Okay? So let's think about this. You start out with spermatogonia, which are true diploids. They're constantly dividing by mitosis. As you get closer to the bridge, signals tell it to undergo the first DNA duplication. You get a primary spermatocyte, which is 46 chromosomes, 4 cDNA, goes through, in between those Sertoli bridge, on the inside of this cross-section of the seminiferous tubule, undergoes meiotic division, first one, to get secondary spermatocytes, which is 23 chromosomes, 2 cDNA. This undergoes in the second meiotic division. You get spermatids, which is a true haploid, and these spermatids differentiate into spermatozoa. These are actually immature spermatozoa. Yes, question? Yeah, there's signals. There's a Sertoli bridge stimulate signals. Yeah, not so much the bridge, but the Sertoli cells themselves. And there's different hormonal mechanisms there, but don't need to worry about that. The point is, what you want to make in the middle of the seminiferous tubule is a true haploid that can swim around a little bit, or spermatozoa. And what goes on each side, the outside of the cross-section to the inner part, is different. Okay? Because you want to keep the mitotic events separated from the meiotic events. Basically, is what's going on. Okay? Other questions? Okay. So again, think about it in terms of the DNA, because for the females, it's a very different kind of setup. I'll leave that there. So what comes out of the seminiferous tubules is you have a whole bunch of these ducts, multiple ducts. And these guys are immature spermatozoa. What happens is they get into a single duct called an epididymis. So it's a single duct, single large duct. And in this span, from here to here, it takes about 20 days, including storage, and the spermatozoa mature. So these are now these spermatozoa can swim around. And what the mature spermatozoa look like, kind of like this. There's a front part called an acrosome, which has enzymes to digest. Call it the egg, female egg plasma membrane. We'll get to that certainly by Friday. There's the nucleus. Okay, the nucleus is 23 chromosomes cDNA. You have a tail that likes to move and needs energy. There's a whole bunch of mitochondria. So it's basically a mobile nucleus is what it is. Okay, this is